Hello, everyone. Good morning, good afternoon, good evening. We're all around the world. Today, we're making history together for the first time. We are coming together, people from 22 countries, to recognize Neuroblastoma Day, a global initiative dedicated to raising awareness for the rare and devastating childhood cancer disease. This is more than just a day on the calendar. It's a united effort to shine a light on our children, family, medical professionals who face neuroblastoma every day. It's about raising our collective voice to demand more research, more understanding, and better access to information and resources for everyone impacted by this disease. The creation of Neuroblastoma Day reflects the importance of coming together as a global community. By uniting parents, caregivers, advocates, and experts, we're building bridges between the people living this experience and the knowledge that can make all the difference. Awareness might save lives, and access to the right information, whether it's about cutting-edge research, new treatment options, or family support, it's critical to ensuring that no one feels alone on this journey. For me, this initiative is deeply personal. I founded Shir for Life in honor for my youngest son, Shir. Shir passed away on December 5th, exactly 3 years ago, and today, this day is for all of us. Shir's story is one of love, resilience, and unbreakable bond between a parent and their child. His memory fuels my determination to ensure that no family has to walk this path without the support and resources they need. I want to express how honored I am to be part of today's International Neuroblastoma Summit, which brings together the world's leading experts in neuroblastoma. We're privileged to have here today Dr. Susan Kohn, Dr. Roba Gatel, Dr. Julie Park, Dr. Lucas Moreno, and Dr. Maya Beck-Popovic. Their work is transforming the landscape of neuroblastoma research and care, and their insights today will help us better understand how to break boundaries and drive progress forward. On a personal note, I want to thank a few people who were critical for the success of this event. Dr. Judy Villablanca from the NAND Group, Nicole Scobie from Zoe for Life, Nick Bird and Leona Knox from Solving Kids Cancer, Susan Jostoff from NAND Parent Advocacy Group, and Priscilla Ma for Band of Parents. Now, I would like to hand over to introduce you to Dr. Maya Beck-Popovic, pediatric oncology in Lausanne, Switzerland, involved in SIOPAN since many years, currently the past president of SIOPAN. Thank you, Maya, for being with us today and for your contribution to the neuroblastoma field.
Thank you very much, Einat. Thank you for this great and very important initiative, for giving me the opportunity and honor to participate and to be part of this first International Neuroblastoma Awareness Day. As you have said, we have over the year moments of awareness for childhood cancer, and neuroblastoma is certainly among the diseases that merits particular attention. We know that progress has to be made, not only for patients that have high-risk neuroblastoma, we also have patients that should do well and do not well. So a lot of work still needs to be done for all these children. I'm very happy to be part of this honorable committee with so many experts in the field. And let me start by introducing Dr. Susan Kohn as our first speaker. It is a big pleasure, Susan, to introduce you. Dr. Susan Kohn from Chicago. Dr. Kohn has been a teacher and a model for many pediatric oncologists throughout the world, and has devoted her career to caring for children with neuroblastoma and conducting clinical and translational research in order to understand better biology and to help to identify new therapeutic targets. Dr. Kohn is currently Director for Faculty Development at the University of Chicago's Comprehensive Cancer Center. Among many awards received, I would like to highlight the 2022 Lifetime Achievement Award from the Advances in Neuroblastoma Research Association. Susan, we are looking forward to listen to you.
Thank you so much. It's truly an honor to be here, and I'm just going to pull my slides up real quickly. Whoops, they didn't come up. Okay. So it's truly an honor to be here, and I was asked to give a little summary of a project that actually I've been working on with many, many colleagues from around the world, including my co-chair, Dr. Andy Pearson from the UK, developing the International Neuroblastoma Risk Group. And this is as I will describe, has really been transformative, I think, in terms of being able to accelerate research and international collaboration. So way back in 2004, Andy and I were asked to develop and build an INRG task force. And we had the following goals. We recognized that in North America and in Europe and in Japan, different risk group schemas were being used to direct therapy. And or high-risk patient across the world was not the same. It was impossible to compare the results of clinical trials. So we had two major goals. One was to establish a consensus approach for neuroblastoma classification, which we hoped would accelerate our ability to interact more internationally. And then, of course, also wanted advanced research that would improve the outcome of children with neuroblastoma. So our first goal was to develop this pre-treatment risk classification system, and we brought everybody in the room. What we ended up doing was a statistical analysis of 36 different prognostic markers that were being used across the world. And we were able to collect data on 8,800 patients from around the world who were enrolled on various clinical trials. This large cohort had previously been unheard of. It's really an unprecedented cohort of neuroblastoma patients at the time. And that's because neuroblastoma is a rare tumor, and there have never been large cohorts of patients before that have been collated. So we did our analysis with the help of Wendy London and all the others on the task force, and we were able to develop a INRG risk classification system. And we also worked with other folks on the task force to develop a pre-treatment INRG staging system, because we wanted this to be a pre-treatment INRG classification system. And both of these papers were published now almost 15, 16 years ago. I just thought I'd show you what this risk classification looks like. We use INRG Stage, which is an image-based staging system. We use age as a stratification marker. Histologic category, there's different kinds of neuroblastic tumors, including tumors that are called nodular ganglioneuroblastoma or immature neuroblastoma. We used grade of differentiation, the presence or absence of NMIC amplification, and we also included a abnormality of a chromosome 11q as one of the prognostic factors, and ploidy. And if you can just work across this, and if a patient had these individual characteristics, they were then categorized as either very low, low, intermediate, or high. And then the various cooperative groups could use these risk classifiers to then assign patients to clinical trials in their groups. Now, the other thing we recognize is that this 8,800 patient cohort was really quite remarkable. And we felt that this large cohort of patients should be available to the research community, and we wanted to share the data. And that was really a huge impetus for in terms of making sure that all stoma. And indeed, we were able to take a look particularly at very rare cohorts of patients. So for example, patients who have metastatic lung disease, it's a very rare cohort, never really could have been looked at with smaller numbers at individual cooperative groups. And when we put all these patients together, we were able to identify those patients and analyze those patients. We always also did in fact, Maya and Ro and I worked on a very rare cohort that was looking at patients who had localized tumors that were NMIG amplified, also a very rare cohort. We've looked at adolescents and older patients, and then we've looked at a very unusual metastatic spread of disease, pattern of disease, which is classified as stage 4N, which is just disease that's solely metastatic to lymph nodes. So these are really important papers that never would have been possible without this large cohort. Then in 2013, there was another transformation. I, at the time, was working at the University of Chicago, and I collaborated with a mathematical professor in the Department of Mathematics, whose name was Bob Grossman, and also Sam Vochenbaum, who was in our section of hematology oncology. And they were able, Bob really had the expertise, and Sam really spearheaded this effort to develop a data commons. And Bob had been developing a data commons actually for all the genomic data that was being generated by the NCI, and the University of Chicago houses the NCI data commons. But we wanted to develop a data commons that would house our neuroblastoma data. And this is cloud-based, and there are analytical tools within the clouds. And what you can do is you can actually upload new data, so new cooperative group data from all over the world was added to the iNRG database. But what's quite remarkable is through APIs, the data, the clinical data can be linked to external data sources, including the NCI Genomic Data Commons, which does contain both adult and pediatric genomic data, as well as the Gabriella Miller Kids First. And we also have a small cohort of patients that are linked to the GEO database that has some SIAPIN data on some localized patients. The researcher can then link into the data portal and do a cohort discovery to see how many patients are present that meet their particular criteria. And the researcher can also receive patient-level data to do applications and research projects through an application process. So this is our website, the iNRGDB.org, and on this website, there are a variety of different links, and I thought I'd just real quickly through this cohort discovery tool. So if you link on this tool, you get sent to the Pediatric Cancer Data Commons, which really was leveraged from the work that we did in neuroblastoma to now have other pediatric cancer data under this umbrella. So what we started has now been expanded to a variety of other pediatric cancers. But if you specifically want to take a look at the neuroblastoma data, you can see you can just check the iNRG, which holds all the neuroblastoma data, and you can now see we've got 25,404 patients in our database, huge increase from the 8,000 we collected previously. And here on the first page, you can get some information about their sex, their race, their ethnicity, and the consortium in this case is the iNRG. And then as I mentioned, it's also possible through this cohort discovery tool to link on the cohort that you're interested in, and then to see if indeed there's genomic data available in NCI Data Commons. And this just shows you how you can link that. Once you link it, you get an idea of how many patients there are that have genomic data available. And then you can actually upload the case set and do some deep dives on the genomic data if you get to access the genomic data site. Now, with this data, both the clinical data as well as the links that we have, we've been able to do a number of different research projects. And again, you can link right onto this research tab, and it brings you up all the different studies. These are more recent studies that have either just recently been approved or are in progress. But there are also older studies that are actually linked to the actual publication, and you can pull up the PDF of the publication if you're interested. Now, we're not done yet. Although this is very exciting, as we've been doing these studies, we're very aware that there are some limitations to the data that we've collected, which have limited to some extent the types of studies that we can do. And I've been working with folks in Sam's group, particularly Brian Ferner, to try to figure out how we can enrich the data. And there are all sorts of new technologies that have been developed to be able to add data from electronic health records. In the more recent era, all our patients have electronic health record data. And we've recently completed a pilot project utilizing a new way of extracting electronic health record data. And we specifically focused on treatment data. And we've successfully done this now for 2 different institutions. And we've actually added the actual treatment data to the iEnergy database from our University of Chicago patients that were in this particular pilot study. And we're about to now add Vanderbilt treatment data. So you get the actual drug that they were treated with, whether they got anti-GD2 antibody, when they got it, the date they got it, et cetera, et cetera. So it'll be very detailed now treatment data. And we're hoping to add additional electronic health record information about toxicities and response as this project expands. We're also increasing some genomic data that are actually housed in the iEnergy, not linked to the databases, but rather housed in the iEnergy. And again, what we've done already is we've added some data about ALK mutations. I think many of you on the at this meeting recognize that ALK is one of the most common mutated genes in neuroblastoma, and that there are now drugs that will target ALK if indeed you have an ALK mutation that appear to be effective. In addition, we've uploaded some digital medical imaging, including MIBG scans, and there's future plans to upload some digital pathology data. And there's been at least one study that's been done taking a look at some of these MIBG scans that were uploaded and using AI and machine learning to try to understand how the MIBG scan at diagnosis might be able to provide some information about the ultimate outcome of the patient. And then, of course, we're very interested in increasing our connection to external data sites, and we're working diligently with a variety of other groups that have genomic data to expand these connections. So in conclusion, I believe the iEnergy Task Force has transformed international and intercontinental collaboration and communication by forming a framework for investigators in different countries. The classification system has enabled a consensus of risk-based therapeutic groupings, and we now are able to compare treatments across the world. And the data commons has enabled high-impact research studies, catalyzed international collaboration, and led to harmonization of data that includes staging biomarkers and risk classification. And sharing expanded data, enriched data in the iEnergy will advance research studies and lead to new discoveries that we hope will ultimately improve the outcome for children with neuroblastoma. And I just want to acknowledge my colleagues at the PCDC and my colleagues on the iEnergy Task Force and all the investigators. And certainly the parents and the patients who have enrolled patients on our clinical trials and have enabled us to have all this data to do these research studies, as well as a number of different foundations that have supported this effort. Thank you very much.
Thank you very much, Dr. Kohn, Susan. Thank you very much for this very clear presentation. So we have seen that the database is extremely rich in information. A lot of information is there on many patients, and certainly we have a lot of work in front of us. You have very well shown the studies and publications where the data have already been used, and there is much more to be done in future. So there is a question from a participant, and it is a cohort question. So it is from a father questioning for the high-risk stage 3 neuroblastoma patients, the role of bone marrow transplant, high-dose chemotherapy and bone marrow transplant. It is part of high-risk treatment, usually presented as something needed in order to give an adequate treatment to this type of high-risk patients. The question is this still true? Is it the only option for stage 3 high-risk patient, as it is usually presented at the first discussion at diagnosis, or other options available? Do we have an answer to this question, or is this something that can, through the INRG database, be evaluated and explored?
I'm going to turn this over to some of my other colleagues who, like Ro and Julie, but I do want to say that we're well aware that in Europe, the approach to high-risk stage 3 can be a little bit different than in North America. And there are a number of different investigators who have tried to compare results from clinical trials. In the INRG per se, I think once we get some more enriched and detailed treatment data, that might be something we could also look at using that using that data commons. But at this point, we don't know exactly how individual patients, we know who's stage 3, but we don't know exactly how they were treated. And that's what I was saying, that there are some limitations to the types of research studies that can be done with the INRG data as it exists today. But I know that the details of treatments are available on the therapeutic trials, and maybe Dr. Bagatelle or Dr. Park might want to address this further.
Do you want to make a comment to that?
I wonder if we should talk about this a bit more after our the presentations, because some of the points that I think we'll make will help to clarify this.
I think this is a good option because we have another, so this is not forgotten. We will discuss it later, then maybe after your presentation. There is another question related also to energy. How do these data feed therapeutic decision? Do pharmaceutical companies or trial investigators have access to these data sources in order to make decisions for trying new drugs?
So right now, these are strictly risk-based, treatments that we've been doing. Again, I think that most of the if they're experimental pharmaceutical trials and not upfront clinical trials, those patients are typically patients who have relapsed disease. Or a disease that hasn't responded to upfront therapy. And the focus of the original database was largely collecting data from upfront newly diagnosed patients, although we are now beginning to start to add some relapse patients. And so there, I'm sure we'll end up learning new things about patients who have relapsed in various biomarkers perhaps that will be relevant as treatment decisions are made about new treatments.
Yeah, thank you. Thank you. And maybe one more question, and then we will have to switch to the next presentation. This is the role of artificial intelligence and how is it being used in research and review of the patient data? So this is a big question in all the fields currently.
Yeah. Yes. So as I said, there have been individual projects where AI has been used in the INRG, and particularly in this one project looking at the images, the digital images of MIBG scans. There are other projects that are ongoing, also looking at the path using AI and machine learning. That hasn't yet been done through the INRG data commons because we don't have that path data in there yet, but that is a goal. It would be to add some of the pathology data. I do know, though, that within there are some studies that are ongoing, taking a look using AI and machine learning at neuroblastoma pathology to see if there's a way to kind of utilize it, much like it's been done with some of the adult cancers. And it's also been used, for example, in mammography for breast cancer. So similar kinds of things have been done with AI, and it's being just now beginning to be looked at in pediatric cancer and certainly in neuroblastoma.
Thank you very much. I don't see other questions. So Susan, thank you again.
My pleasure. Thank you so much for inviting me to this.
And we will switch over to the next presentation. So our second speaker is Ro Bagatelle from the Children's Hospital of Philadelphia, also highly awarded for her career, excellent teacher, which is reflected in the Top Hematology Oncology Fellowship Teaching Award recently. Dr. Bagatelle has significantly contributed to improving treatment and outcome of patients with high-risk refractory relapsed neuroblastoma. It was also a very big pleasure for me to collaborate closely with Ro Bagatelle, who is chair since 2019 of the COG Neuroblastoma Committee at the same moment and during the time I was SIPEN president. So it was a big pleasure to share and to have a lot of activities in common. So I'm looking forward to your presentation, Ro, please.
Thank you so much, Maya. It really has been a pleasure to work with you over the years. And Sue's presentation reminded me that we go way back to that first paper that we did together and with Sue. So it's great to be here with you, and really amazing to be here with all of the people in cyberspace. I'm just really impressed with the attendance at this, and I'm so glad that you took the time to join us today. I will share my slides. I hope people can see that, okay? I'm gonna say, unless I hear otherwise, I'm gonna think it's okay. All right.
Ro, it's swapped.
It's swapped.
We're seeing the presenter mode. If you, this happened to me yesterday, if you go back out and then put it in presenter mode first, and then
Okay.
try again. It worked for me yesterday.
Can you see my slides?
Yeah, it's not in presenter mode.
Can you get your mouse to the top? There's a thing here that says swap displays.
Yeah, you should click swap displays on the top
You
left.
see it to the left here?
Yeah. Okay, great.
Okay.
There
All
you
right,
go.
thanks, everybody.
Perfect.
Okay, great. All right. Well, thank you very much for all your help in this nice neuroblastoma community. I'm here today to talk a little bit about the Children's Oncology Group, which is the North American cooperative group that also includes a few other countries around the world. The majority of the institutions are in the United States and Canada, but we do have participating centers in Australia, New Zealand, and Saudi Arabia. And altogether, there are 226 institutions that participate in Children's Oncology Group trials. The Children's Oncology Group was founded in 2000 after the merger of two previous cooperative groups based in North America. And I think what happened after that merger was that the strength of both groups was really combined, and the number of institutions and patients was increased so that we could conduct really critical trials to answer pressing questions in the field. About 600 new patients with neuroblastoma are enrolled on the main COG biology studies per year, and subsets of those patients then go on to our therapeutic trials as well. I've shown here the pictures of some of my leadership team. Meredith Irwin from the Hospital for Sick Children in Toronto is the biology chair for the Children's Oncology Group Neuroblastoma Committee right now. Steve Dubois from Dana-Farber is the overall vice chair, and Kelly Goldsmith from Children's Healthcare of Atlanta is the biology vice chair. We work very closely on a variety of activities that I'll talk about in just a few minutes. I'd like to acknowledge my predecessors, though, a couple of whom are on this call. So Julie Park was the COG neuroblastoma chair just prior to me and taught me most of what I know about how to get along and get things done in COG. And of course, Sue was the first COG neuroblastoma committee chair back in 2000. The COG neuroblastoma committee leadership has met to really try to frame our mission, and that is to conduct practice-changing trials that improve outcomes for our patients. And when I say practice changing, I really mean primarily in the frontline and first relapse setting. So COG does conduct early phase trials, but primarily the strength of COG as far as neuroblastoma goes is to really test hypotheses on a larger scale and influence the practice that takes place at the 226 institutions that are members. In order to achieve this mission, the strategy that we have selected is a strategy of engagement and inclusion. So we really want to encourage investigators with all kinds of different expertise from across the range of COG centers to participate in our trials, lead our trials, ask pressing questions, and really get involved in the group, whether you're from a small institution with 40 new patients per year or a large institution with more than 300 pediatric cancer patients per year. Everyone's experience matters and everyone's participation really makes a difference. And that I feel is very important. And I think there's a consensus about that because the patients come from all kinds of institutions and we really want to focus on improving outcomes for everyone. The other component of our strategy of engagement is collaboration with colleagues from around the world. And certainly I had the honor and privilege of working very closely with Maya over the years, and now with Lucas and all of our colleagues from ScioPen and from other cooperative groups, so that we can exchange ideas and not necessarily repeat the studies that are being done by one group or another, but actually learn from them and push forward faster. And then another major component of our work is to partner with our COG neuroblastoma parent advocate as a representative of other parents out there, and with other parent advocates from other groups to try to understand what it's like for parents in a way that we could never understand necessarily ourselves. So engaging parents is really crucial, and I'll talk in a minute about one very important way that we're pushing forward based on the suggestions of parents. So the framework that we have put into place is to really put emphasis on inclusive biology and biobanking studies, trying to collect as many specimens as possible from patients diagnosed with neuroblastoma and then study them as thoroughly as we can. We do include biobanking of patients with non-high-risk disease as well as high-risk disease. And we've recently been able to take advantage of the molecular characterization initiative to further sequence and understand what's happening with the tumor specimens that we're collecting. The other thing that the COG Neuroblastoma Committee does is to conduct frontline pilot studies, primarily for patients with high-risk disease. And those are mainly designed to test the feasibility of new interventions that may be further studied in larger randomized trials. The randomized trials are really one of the major strengths of COG, the best way to test hypothesis and to really get strong data that investigators, clinicians, and parents can believe is to ask a question about a treatment and have half the patients get the standard therapy and half the patients get the new therapy, and be able to really say with confidence, based on a randomized trial, whether the new therapy improves outcomes. And I'll come back to that in a minute. We do run trials for patients with relapsed and refractory disease, but generally in the setting of first declaration of refractory disease or first relapse. And we have a nice track record for making some new therapies move forward into the frontline setting based on our first relapse studies. We primarily partner with NANT, and Julie will talk about the discoveries that can happen in a primarily new agents focused consortium that can develop drugs and study new treatments in patients with multiply relapsed disease, whereas COG has focused really on the first relapse setting. As we conduct this variety of trials, we really emphasize correlative biology because the patients are treated with the same therapy on our trials. It's a uniform therapy. We know the details of the therapy, and we can then use biospecimens to understand what's happening during the course of therapy in the tumor cells and also in the host cells as well. This gets at one of the issues that Sue was talking about, where the INRG initially did not have as much detailed information about treatment received, whereas our therapeutic studies are really strengthened by the fact that patients are uniformly treated, and we have this robust collection of biospecimens. COG also conducts what we call companion studies, looking at ways to decrease the toxicity of our therapy. So for example, hearing loss is a major side effect of current high-risk neuroblastoma therapy, and we're currently pushing forward a concept to try to look at ways to decrease ototoxicity in patients. We're also interested in making better decisions around time points where we need to switch therapies. And there are a couple different ways that we're looking at disease assessment, both with circulating tumor DNA, and that testing has been incorporated into our frontline trials, as well as new imaging technology. So we have a clinical trial that the concept is being reviewed right now, looking at a new type of imaging study that we hope someday could replace the rather onerous testing using MIBG scans. We also conduct trials for patients with non-high-risk disease. And I think as investigators and often as parents engaged in the neuroblastoma community, our focus is often on patients with high-risk disease, but fully half of patients diagnosed with neuroblastoma have non-high-risk disease, so low or intermediate risk disease. And we do want to continue to improve outcomes and decrease toxicity for those patients. So that's a lot of work, as you can see. I want to just explain a little bit of the concepts that I talked about. So practice-changing trials are really what we aspire to do. And one of the ones that has been a big success for COG was this ANBL 0032 study of immunotherapy after high-dose chemo with stem cell rescue in patients with high-risk neuroblastoma. This trial was conducted by Alice Yu and colleagues at the beginning of this century, but the publication really was instrumental in making anti-GD2 immunotherapy more widely available, both across North America and increasingly across Europe and other continents. And you can see the 20% improvement in event-free survival in patients who received immunotherapy compared to those who did not. So that's the kind of trial that we really want to conduct. The second example of this kind of practice-changing trial was conducted by none other than our colleague Julie Park, who's right here. This was the ANBL 0532 study, where half of the patients got a single cycle of high-dose chemo with stem cell rescue, and the other half of patients received back-to-back cycles of high-dose chemo with stem cell rescue. And what Julie and her colleagues found was that there was an improvement in event-free survival for patients who got the back-to-back high-dose chemo with stem cell rescue. After that trial, tandem transplant or back-to-back transplants became the standard of care in North America. So really changed what we were doing. And those are the kind of studies that COG is set up to do. An example of a paradigm shifting first relapse study was a trial that we ran a number of years ago with a chemotherapy backbone of irinotecan and temozolomide with two different categories of targeted agents, with the idea being that there was a modest response rate to the chemotherapy alone, but the addition of targeted agents was expected to improve response rates in patients with first relapse disease. We had no idea when we started this trial that we would see the kind of response rates that we ultimately did when we combined irinotecan and dinutuximab and temozolomide together. And you can see in the chart at the bottom of the slide that the objective response rate, complete and partial response rate, was just over 40%, which is not 100% by any means, and certainly we need to improve this, but 40% was a response rate that we had never seen before in patients with relapsed and refractory disease. And this, together with some data from our colleagues at St. Jude, combining chemotherapy and immunotherapy, really was quite exciting to us and motivated us and others to push forward with chemoimmunotherapy in the frontline setting. And so with those data in hand, we then conducted a pilot study to determine the feasibility of administering immunotherapy along with induction chemotherapy shortly after diagnosis on a multi-institutional level. The different institutions that participate in COG have different resources, different contexts, and so it was really important to us to move from the relapse setting and also beyond the single institution setting and see if it was practical to give chemoimmunotherapy early on in treatment. And indeed it was. Sarah Federico and her colleagues led this study and nicely presented these data at an international meeting relatively recently and showed that we could push forward and study this in the frontline setting. Another example of the kind of work that comes out of our first relapse study and the finding that about 40% of the patients responded was the idea that we need to improve on this and increase that number so that it's really the majority of patients who respond. And one of the ideas for the ANBL 1821 study was to use irinotecan, temozolomide, and dinutuximab, but have half the patients receive, in addition, the ODC1 inhibitor DFMO, otherwise known as eflornithine. People deep in the neuroblastoma community, of course, have heard of DFMO, and this was approved by the FDA for use in a different setting. But based on some work by my colleague Mike Hogarty and others, there seemed to be modulation of the immune function associated with dinutuximab when DFMO was also included in the regimen. And this study has now been completed. And I think we can really learn a lot from this about how to augment the activity of chemoimmunotherapy. Meg Macy and her colleagues have run this trial, and we hope that these data will be presented at ANR this spring in Washington, DC. The other activity that COG is well set up to do is to really take biology studies conducted using our large tumor bank and add components to clinical trials based on rational design and what we learn in the laboratory and bring it over to our clinical practice. So Yael Mosé and others did some really important work about a decade ago looking at the anaplastic lymphoma kinase gene and focusing on mutations that are seen in neuroblastoma or amplifications, too many copies of the ALK gene, and seeing how that impacted outcome in patients with different stage disease. And what Dr. Mosé and her colleagues found was that patients with aberrations, either mutation or amplification in the ALK gene, had inferior outcomes compared to patients whose tumors did not have changes in the ALK gene. And that was really the impetus to incorporating a specific arm for patients with ALK aberrant neuroblastoma in our large phase 3 clinical trial ANBL 1531. The main part of this study was for patients whose tumors did not have an aberration in the ALK gene. And the question on the main part of this study was whether early inclusion of MIBG improved outcomes in patients newly diagnosed. But importantly, based on the work that Dr. Mosé and others had done in the laboratory, we included this arm over here, which incorporated first the ALK inhibitor crizotinib and later the ALK inhibitor lorlatinib into frontline therapy. So this really sort of suggests that the paradigm of biologic discoveries using the COG biobank and further development to bring it forward into clinical practice is really what we want to be doing. I will note that lorlatinib was first studied in the NANT consortium and showed promising results, which prompted the change from crizotinib, the first ALK inhibitor, to lorlatinib, the more recent ALK inhibitor. Building on that concept of correlative biology that I mentioned earlier, this large study, ANBL 1531, conducted primarily to answer the question about the inclusion of MIBG in treatment, but also including the lorlatinib arm, was a pretty large study when you think about neuroblastoma trials, and more than 700 patients enrolled. What we committed to at the time of inception of this trial was to really try to strengthen our correlative biology studies and learn as much as we can from every patient that was enrolled. And so we included a whole panel of correlative biology studies with basically an outline, as you can see here, drawn by Meredith Irwin and Kelly Goldsmith, to look at the tumor immune microenvironment, ctDNA, to look at telomere maintenance mechanisms, gene signatures, and GD2 on tumor cells, as well as host characteristics, to really try to understand what was happening as we treated our patients. My automatic light is going down. And so those studies are ongoing. We've studied quite a few patient samples already, and a few of those early results will be presented also at ANR 2025. We don't have the clinical outcomes from this trial yet, so we can't really publish the full data, but we're starting to get some intriguing early results from these biology studies. I just wanted to give you a brief update of what we're doing currently in the COG. Our main high-risk study that is open and accruing patients right now is called ANBL 2131. And this is a randomized study. Half the patients get standard therapy, as shown on the top line here, a multi-agent chemotherapy and surgery induction, followed for patients who have a good response by back-to-back stem cell transplants and then immunotherapy. And that is being compared to standard therapy plus the addition of dinutuximab, an anti-GD2 antibody. This builds on both the relapse study that we conducted about 10 years ago, also on the pilot study that showed that it was feasible to administer this regimen. And now we're studying it at over 200 institutions to determine whether this approach really works and can be relied upon or not. In the future, our next step is going to be ask another really pressing question in the field. And this is the one that I think was really pushed forward by parent advocates. They spoke and we listened. So there was this first article in the medical literature by parent advocates published by the group here led by Nick Bird and others, where they published in a medical journal, Pediatric Blood and Cancer, kind of a position piece, very well written, saying how important it was to really answer the transplant question definitively and not just based on retrospective studies or practice patterns or anything else. So what we would like to do in the COG is take our induction regimen and take those patients who have a good response, either an initial good response or an augmented response with chemoimmunotherapy, and have the patients get our conventional therapy demonstrated to be superior based on the 0532 results, and have the other half of the patients not receive transplant, but instead receive additional chemoimmunotherapy to be able to rigorously answer the question for patients, for parents, and for providers, what do the data tell us? Not opinion, not anecdotal evidence, but should we be transplanting patients or should we not? And within those groups, are there subsets of patients who really benefit from transplant? And are there others who could be spared the toxicity? So that's our plan going forward. That will be our next big trial. And again, that was really pushed forward, and the importance of conducting this in a rigorous way was really advocated for by parents. We continue to want to improve relapse therapy. We think that chemoimmunotherapy is really a big step forward, but as shown here, this is an imaging study of one of my patients with a very large tumor that just progressed right through chemoimmunotherapy. So we really need to augment the effect of our chemoimmunotherapy, potentially by using additional agents, perhaps something like DFMO or other agents of that nature. Maybe we need to use a different anti-GD2 antibody. Maybe we need to have a different approach to target GD2 or other key molecules. Maybe we need to improve our patient selection. So if we identify biomarkers that can tell us, hey, this patient is going to respond well to chemoimmunotherapy while this patient is not, we could select our treatment better. So we're very much interested in trying to improve the effects of chemoimmunotherapy and introduce something new even into the frontline setting. As I mentioned, we are interested in toxicity reduction, and I mentioned the fact that we are putting forward an ototoxicity prevention study. And we're also interested and have proposed a new imaging study using a tracer called MFBG instead of MIBG. And some of the beautiful pictures are shown here. This is an MFBG scan showing something that wasn't picked up on an MIBG scan. So hopefully that will be happening in the future as well. I do also want to just highlight the very important work that Brian Crompton and others have been doing regarding improved decision-making with circulating tumor DNA. So much more to follow on that in the future. And other important work, as I mentioned, to try to develop predictive biomarkers for our immunotherapy studies and other biomarkers to guide our decision-making as well. The strength of the COG Neuroblastoma Committee is that we have the patient numbers to be able to rigorously study these things and learn what's helpful and what is not. So I'll stop there. I'd just like to thank all of you for listening today, and also the many, many people that it takes to design, implement, and run COG trials. It's a huge undertaking. COG is a complex, multifaceted institution. Sometimes, all the time, it moves too slowly, but we really can get important work done, and it's due to the tireless efforts of many, many, many people. So I will stop there. And I guess, Maya, turn it over to questions.
Yeah. Thank you very much, Ro. First, thank you very much for this very clear and exhaustive presentation. And it really encompasses many aspects. You have very well shown also how concerns by parents can be translated into research questions that will help to answer these concerns, and also the need for long-term follow-up. But one thing is to reduce treatment, to reduce toxicity, but we have also to follow the patients on the long term. And this is also something that in Europe and US exists, the long-term follow-up of neuroblastoma patients. As your presentation, Julie's presentation, and Lucas are very much linked together, maybe we go through the presentations and have the questions that are coming up discussed at the end. Is this fine for you, Ro? Einat, is this okay? Good. Then let me introduce Julie Park. So Julie Park from the Department of Oncology at the St. Jude's Children's Research Hospital, name and personality that stand for innovation, clinical application of laboratory discoveries, development of early phase trials for recurrent malignancies, especially of immune-based therapy. And with her expertise as the scientific chair of New Approaches to Neuroblastoma Therapy Consortium, the NANT consortium that has been mentioned, whose goal is to translate laboratory discoveries into novel therapies for children with neuroblastoma, Julie Park aims at accelerating translational research by accelerating the development of novel clinical trials derived from laboratory projects and their results. So we are very interesting to hear your presentation, Julie. Thank you.
Thank you, Maya, and thank you everyone for pulling this together and allowing me to participate in this really wonderful session. Let's see if I can now also. Am I having the same problem as Ro, or do we have the full screen?
No, you've got to switch it. You got to swap it.
Okay. Well, again, thank you very much. Is that better? Great. Well, as Maya said, I'm here to talk about the new approaches to neuroblastoma therapy. Ro has mentioned this a few times in her talk, and I'm very privileged to be the scientific chair for this consortium. The NANT consortium was really founded in 2000 and is the brainchild of my prior mentor, one of my prior mentors, the other one's on this call, Kate Mathey, working very closely with Judy Villablanca. And they developed an operations center and infrastructure to run early phase clinical trials for children with relapsed and refractory neuroblastoma that was based at Children's Hospital of Los Angeles. And this was really at a time when less than 50% of all children with high-risk neuroblastoma were surviving from frontline treatment, and really no treatment for relapse and refractory neuroblastoma was found or was available. And so really they had the forethought that we needed to come together to work on this very difficult to treat refractory and relapse neuroblastoma, and that the hope would be that our learnings would then help to define and help to guide how we might treat patients in the upfront setting. I now have the privilege of partnering with Araz Marichelian, who took over from Judy Villablanca as medical director of the NANT Operations Center. And of course, we still have great guidance from Kate and Judy. The NANT Consortium is an international consortium. It started as North America only, has 14 institutions within the United States and one in Canada at Toronto Sick Kids. We've now expanded to 4 sites in Europe, in UK, the Netherlands, United Kingdom, France, and Germany. The NANT consortium is the first consortium that was solely dedicated to innovative clinical trials for relapse and refractory high-risk neuroblastoma. And I believe it is still the only consortium that really is focused on just this group of patients. Since the inception of 2000, we've treated over 800 patients on early phase clinical trials, and over 650 additional patients have enrolled onto a biobanking trial, developing a resource for investigators who want to study relapsed and refractory neuroblastoma. Currently, we have only 2 active trials. We range from 1 to 6 at any one point in time, and this really depends on availability of novel agents that we can get for clinical trials, and sometimes the complexity of the clinical trials that we run, and whether how many we can support at any one period of time. So I'd like to just quickly review some of the accomplishments of the NANT consortium over the past 20, now almost 25 years. Ro actually mentioned many of them, so I thank Ro for that. As I said, the goal of NANT is to accelerate the translation of laboratory findings into novel clinical trials for relapse and refractory high-risk neuroblastoma. And we do that by partnering very closely with laboratory scientists and reviewing their results and translating those results into clinical trials. We do that first through an NIH grant. That's a program project grant that brings together 8 laboratory-based investigators with the NANT consortium to try to study mechanisms of resistance of high-risk neuroblastoma and then target those into clinical trials. But we've also expanded now in working with investigators, laboratory investigators in Europe as well, so that we have as much information as possible to translate into clinical trials. And then, of course, we want to establish and maintain an infrastructure that can take this information and rapidly implement it into an early phase clinical trial. And our successes have, as I said, been outlined by Ro. Our early studies in the early 2000s really provided the rationale for studying whether radiolabeled MIBG in the upfront setting will improve survival for children with newly diagnosed high-risk neuroblastoma. And we anxiously await the results of the COG trial. As Ro mentioned, our phase 1 trial of lorlatinib in patients who have high-risk neuroblastoma with ALK aberrations embedded into their tumor showed really encouraging response rates and tolerability. And that allowed us to fast forward and incorporate lorlatinib into the upfront treatment for high-risk neuroblastoma. That data is also was shared openly with our SIOPN colleagues so that they too could have a trial where we can begin to look at lorlatinib in the upfront setting using the backbone of chemotherapy, induction chemotherapy utilized in Europe. And finally, as Ro pointed out, an early phase clinical trial of DFMO in combination with chemotherapy that was performed in the NANT really provided the background data that allowed us to run the recently completed COG 1821, looking at whether DFMO will improve the outcome of chemotherapy when combined with anti-GD2 antibody. And then finally, over the last 10 years, we've really been able to develop the processes necessary for biopsying tumor tissue so that we can obtain really detailed genomic and molecular and immunologic data on tumors, such that we now have transferred those biopsy protocols really to other groups so that we can all begin to start to look at this important information. And Ro has nicely outlined some of the very exciting information that we will now get on upfront clinical trials performed within the Children's Oncology Group. So what are we doing right now? What we're doing right now, I kind of tried to bucket into a variety of different topics. Ro nicely showed the seminal paper by Dr. Yu in 2010 that really turned how we think about treatment with neuroblastoma around. There had been work for 2 decades from our colleagues at the Memorial Sloan Kettering, colleagues within Europe, and Dr. Yu and Dr. Sandel that really led us to be optimistic that immunotherapy would improve the outcome for children with high-risk neuroblastoma. But it was that randomized trial that really established that. In addition, it was the trial that we ran combining chemotherapy with antibody that said for the first time we were really seeing sustained responses in the relapse setting. But as Ro pointed out, only 40 to 50% of patients will respond to that. And that's not enough. And we need to understand what are the mechanisms of resistance to that and how can we improve the immune response against neuroblastoma. So one of the things that we've been working on is developing cellular therapies using immune cells from the patients or from other patients that can help with the immunotherapy process. That's a complicated process, and we needed to develop the infrastructure that we could collect and distribute cell therapy products across at least the United States, and now we're expanding into Canada. So with that, we've been able to open a new study looking at NK cells, which are a form of immune cells from donors, not from the patients. So hopefully they'll be highly functional and be combining those with chemotherapy and anti-GD2 antibody. In the realm of MIBG, as I said, we 20 years ago really did the seminal work in showing efficacy of MIBG in the relapse setting. We have now begun to look at combinations of that, thinking of ways that we can further enhance the efficacy of MIBG. And a early phase clinical trial just completed by Thomas Cash from AFLAC and Atlanta Children's Hospital established safety of combining new agents, vorinostat or anti-GD2 antibody with MIBG. And will soon be opening a randomized trial in the refractory and relapse setting to see which of those combinations will be the best in improving activity of MIBG. And then finally, we continue a biologic specimen repository, collecting biopsies, residual biologic specimens from patients who are enrolled on our clinical trials, again, making those available as a resource to investigators across the world. And in future directions, we're embarking on a biomarker umbrella trial where every patient who enrolls on any neuroblastoma trial through the NANT consortium will have serial samples that look at circulating tumor DNA. We'll look at immune markers. We'll look at the tumor microenvironment when biopsies were obtained. And we'll be able to look at those not only in individual clinical trials, but we'll be able to then combine results of clinical trials and look at these markers across these clinical trials with the real hope that we stop having empiric treatment regimens for patients, but become more precise with each of the patients. We're embarking on new studies that we hope will open in the next year that are further enhance the immune system to increase activity of anti-GD2 antibody, and that includes adding IL-15 immune stimulants, and then antibodies that inhibit the immune system's own checkpoints, own way of suppressing the immune system to try to further increase anti-GD2 antibody success. We are continuing to look at other novel cellular therapy products as a form of increasing immunotherapy. And finally, working very closely with laboratory investigators to begin to bring in new novel molecularly targeted therapies and potential combination therapies with lorlatinib. And then our main, as I said, focus is working very closely with laboratory investigators, both through our NCI-P01, which is led by John Maris, by St. Jude investigators, and our EU and UK collaborators, because it's really this information that will continue to drive us in thinking about novel treatments for neuroblastoma. And then finally, on our geographic perspective, as I said, NANT started initially in the United States, moved to one program in Canada, and now we are partnering with EU and UK colleagues to further accelerate our research and enhance access to early phase clinical trials. And currently, as I said, we're at Princess Maxima, Institute of Cancer Research, University of Cologne, and Institute Curie. We've developed monthly joint meetings. We include representatives from the Children's Oncology Group and from SIOPAN. So we make sure that we all know what we're doing and we don't repeat what we're doing, but instead collaborate and accelerate. Our biobank is open for application from instructors, from researchers. And we're working very closely with the international community to continue to understand how we best have same language for response assessment. And finally, the NANT has a very active parent advocacy committee. They are involved in all of our scientific meetings. They are involved in developing educational platforms for parents and patients, and work very hard for advocacy. And it's currently being chaired by Susan Justo. And we're very, very grateful for their interaction with us and their guidance. So the questions that were posed that would hope we would answer are: what initiatives have we implemented to streamline and expedite novel therapies? I think the NANT infrastructure and the operations group has really done a fantastic job with this. Under Judy's leadership and Araz, we have developed kind of global granting processes, global contracting, regulatory processes to try to quickly implement our clinical trials. We have been very open with partnering with biopharma companies in all different ways so that we can meet them in the middle and get access to drugs that we need for children with high-risk neuroblastoma. And then finally, what are the most effective ways for parents and parent organizations to support and advance neuroblastoma care? My one word is involvement. As as Rose said, we listen to what patients and patient families have to say. We really respect your insight into all of this. And it's really together that we have to work to really improve the outcome for children with neuroblastoma. So with that, I'd like to just acknowledge the many funding resources that we have to continue to operate through NAT. But of course, none of this could be done without patients and their families and their bravery in facing neuroblastoma and enrolling on clinical trials. So thank you very, very much for your attention.
Thank you, Julie, for this very comprehensive presentation of NANT activity. I think you depicted perfectly well what is going on and what still needs to be done and can be done. So let's go to the last presentation. This is a presentation through video by Lucas Moreno, who unfortunately could not participate in person here. So Lucas Moreno is the division head of pediatric hematology oncology at the Vall d'Hebron Hospital in Barcelona, one of the largest tertiary cancer centers in Spain, and is currently the president of SIOPEN. Dr. Moreno's fields of expertise and interests are early clinical trials. He is principal investigator of many first-in-child trials, drug development for neuroblastoma, implementation of personalized medicine for childhood cancer into routine care, and then coordinating of the first international neuroblastoma new drug development strategy initiative for identification of targets and drugs that should focus for early clinical trial within the ITCC consortium, Innovative Therapies for Children with Cancer. So let's hear and listen to his presentation.
Hello, everyone. My name is Lucas Moreno, and I am the current president of SIOPEN, the European Neuroblastoma Research Network. I'm a pediatric oncologist based in Barcelona, in Vall d'Hebron Hospital in Spain. I'm really grateful to the organizers for having me giving this brief overview, and I apologize in advance for not being able to participate live. I wanted to give a brief overview of SIOPEN, which is the European, but now has become an international neuroblastoma cooperative group with more than 25 years of history, and that is working under the umbrella of SIOP Europe. We have got more than 500 full members from 37 countries, and 27 of these countries are fully participating to SIOPEN activities. We now have a new structure based on key roles in the organization that I'd like to mention and introduce to you. First, Maja Beck-Popovic, the past president that has made possible all these changes to function as a more modern organization, to the wise advice from Andy Pearson, that is past president and senior advisor of SIOPEN. And then I work with two vice presidents, Cormac Owens from Dublin and Julia Gray from Southampton, on the leadership for SIOPEN. And recently, Carla Aguilera has also joined as the manager for the organization and is leading on the day-to-day running of our group. And I wanted also to mention that for us, the involvement of patient advocates has been a great improvement and advance in our organization. And from the last few years, we have had Nicole Scobie, Leona Knox, and Nick Bird from Soy for Life and Solving Kids Cancer UK being a great help for our institution, together with other patient advocates, but they have been integrated into our groups, into our structure, and into the development of clinical trials right from the beginning and all throughout their conduct. We also have a strategic plan covering different areas like improving the organization, education, research, working with other stakeholders, but with mainly two areas to focus, which are clinical trial development and delivery, as well as data management issues and problems that arise when we want to put together data from biological studies and clinical trials across all our network of 27 countries. This is the landscape of our clinical trials. We have got most of the time we have got a trial for high-risk neuroblastoma open. We had the high-risk neuroblastoma 1 trial open for many years, led by Ruth Ladenstein, and then Dominique Valteau and Claudia Pasqualini developed the high-risk neuroblastoma 2, which is currently open. We also have the pilot studies open, led by Holger Lode, and the studies on low and intermediate risk group that are currently closed, the LINES and ENSG2. For refractory and relapsed patients, we have conducted and have some trials that are recently closed or in follow-up, like LTI, Veritas, or Beacon, and we have got a first relapse neuroblastoma trial open, which is the Beacon 2 trial. And we have, I know it's also important to mention that we have a registry for spinal cord compression led by Shifra Ash and Toby Traher. And importantly, the bioportal, which is a biological registry of all the patients that have neuroblastoma across the network, that Gudrun Schleiermacher has worked enormously hard to develop over the past few years, that is now open for recruitment, and we are now expanding it into the different countries of the network. This is a snapshot of our current high-risk neuroblastoma 2 trial, where we were trying to test a question in induction that is currently being a bit difficult for drug supply issues. We are exploring the role of chemoimmunotherapy for patients with refractory disease, so patients that don't respond so well get temozolomide, irinotecan, and dinutuximab beta to try to achieve a better response. Then in consolidation, we are exploring a randomization between our standard transplant regimen, the BuMel, busulfan and melphalan, and the tandem transplant, which means 2 transplants, which has shown by our colleagues in the Children's Oncology Group to show some improvement. The patients then go on to radiotherapy, and there is a radiotherapy randomization there with an increased dose for the residual tumor for some patients, and then all patients get maintenance treatment with anti-GD2 and cisretinoic acid. At the moment, we are working to incorporate lorlatinib for ALK-positive patients, and we're also working with this pilot study that I mentioned on incorporating chemoimmunotherapy in induction. And then for relapsed neuroblastoma, we have developed, following the first Beacon trial, we have just opened in the UK, the Beacon 2 neuroblastoma trial that has what we call Tier 1, which is a big randomization of 75 patients per arm exploring chemoimmunotherapy, so dinutuximab beta, irinotecan, and temozolomide in arm A versus the combination of bevacizumab, irinotecan, and temozolomide. But this is a platform trial, and the idea is that more arms can be added as soon as they are ready. At the moment, we do need some more work for other arms to be incorporated, but when they are, there will be more options or different options for the patients. And we have also created what we call this Tier 2, which is a way of testing more combinations so that they can be moved rapidly into the trial. We are now, as I said, we just opened it in the UK, and we're now working to open it in another 17 countries, including Australia and Israel. So we're really looking forward to starting the full recruitment for this trial. As I said, one of our main areas of focus is really improving clinical trial options. And covering these 3 areas, first in the front of operations, we know that operations are important and that there are challenges. So we want to bring active combinations or active regimens into the upfront setting, into the frontline setting as rapidly as possible, because that's the way to make it widely available for the network. We are also working to speed up trial opening process and enlarge the network, and we're working with our clinical trials committee, with the different sponsors of the trials, to make sure that because of the challenge of opening clinical trials in a network of 27 countries, this does not add important delays. And we are also working to widen the access to early phase clinical trials across Europe, because we know that there are selected centers in some of the countries that have got an important number, a significant number of trials, still not enough, because there are still patients that cannot access a clinical trial, but there are other centers and other countries that don't have that many options, and we think that's really important for the clinicians, for the patients, and for the families. In terms of trial ideas, we are excited and working in different combinations and therapies. For example, in developing a GD2 CAR T cell trial across CEOPEN in collaboration with our colleagues from Bambino Gesù Hospital in Rome, as well as other CEOPEN investigators that are developing CAR T cell trials. We are working on developing more active chemoimmunotherapy combinations and also consolidation options for the relapsed and refractory patients, new combinations for MABG therapy, for lutetium dotatate and other radioisotopes, the role of haploidentical transplant, so different options to try to see if patients with neuroblastoma that achieve a good response with chemoimmunotherapy could continue this response with a better consolidation. And finally, but very importantly for us, is to continue doing research in neuroblastoma biology. So in order to have good clinical trials, we really have to have the best ideas, a lot of testing in the laboratory to find the best combinations, the best immunotherapies, the best products, the best targeted agents, and then that together with biomarker development and precision medicine are really crucial to bring these options forward. And to finish, I just wanted to touch on these two questions that the organizers put to us. First, we're really working the organization with patient advocates, with national organizations to accelerate the process to open the clinical trials, but also we realize that after a successful completion of a clinical trial, there is a gap in making the agents available for the patients in having the approvals. We have now different forums like pediatric strategy forums and conversations with together with COG, the European Medicines Agency, and the US FDA in order to facilitate and try to speed up the approval for novel therapies. And regarding the best ways for parents and parent organizations to support and advance neuroblastoma care, I mean, for parents, I just wanted to say that really they are doing the best by looking after their children, by working with their clinicians, by asking questions, by asking about clinical trials or new drugs when standard treatment options are limited. So I really encourage want to encourage parents to work with their team of clinicians and ask all the questions that they think they need to get the best treatment. With patient advocates and patient advocacy organizations, we are already working very, very well, but really we do need to work together in all these fronts, in lobbying to make more drugs available from pharmaceutical companies, to facilitate investment in and research in childhood cancers in general and in neuroblastoma in particular, to work together in multi-stakeholder forums with regulators, with the health technology agencies, and then be involved in the cooperative groups at a national level. There are things that can be done at a national level and others at the European level. So we really welcome all patient advocates to be in contact with our patient advocacy group, and there is a lot of work to do. And with this, I wanted to finish thanking enormously the organization and ANAT in particular, and I hope that this is a very successful day, and happy to continue working together in this front. Thank you very much.
So thank you, Lucas, very much. We will transmit the thanks by email, as Lucas is not here. So I would like to thank all the panelists and for the presentators for the presentations. And we are a little bit late on time, but if you agree, we will take 10 minutes to discuss some of the questions that have arrived in the meantime. And I will try to summarize them a little bit because there are like groups of questions. And maybe one question is, how come there are differences in high-risk treatments between COG, Memorial Sloan Kettering, SIOPEN? How are parents supposed to understand this, to manage, how to know what is best for the child? What can we say to that?
I think we'll let Dr. Park have the first crack at that very complex question.
It is a very complex question. I think to start with, I think it's very important for the parents and the parent community, patient community to understand that we are all the investigators in neuroblastoma, I am positive, are here for one purpose, and it's to try to figure out the best way that we can cure children from neuroblastoma, and in particular, high-risk neuroblastoma, since that is the harder type of neuroblastoma to treat. Many single institution studies can sometimes drive practice-changing results, but they usually have to be validated in larger situations, in larger trials, as Dr. Bagatelle was trying to outline. You'd never know when there's some kind of unintended bias of a single institution trial, of how the children were picked for the trial, how the results were classified for the trial, that might not be necessarily translatable to all children with the disease. And so the benefits of the trials that have been performed within SIOPEN and with the Children's Oncology Group is it takes patients from numerous institutions. So we don't really have the opportunity to handpick one or the other, or potentially even be biased. We try to think very hard about how we are inclusive in the patients that are enrolled, and that we study the question in a randomized way. And that has led to kind of the current model of the treatments for high-risk neuroblastoma. And although the drugs might be a little different between SIOPEN and between the Children's Oncology Group, the general phases of treatment are very similar. We all, as researchers, would love to decrease the toxicity of the treatment for patients with high-risk neuroblastoma, and transplant contributes a lot to that toxicity. But we're struggling with the fact that we know from randomized trials that adding this intensive phase of treatment has improved the outcome for children with high-risk neuroblastoma. And I think we all want to get to a point where we have a good enough response to treatment and a regimen like the chemotherapy and immunotherapy together that we think could substitute for a transplant. But then it has to be studied in a very close way so that we really can tell families this is an okay way to move forward. I think, you know, it is very difficult for families trying to understand the two options. And I don't think anyone can really tell you at the very beginning of treatment which is the best treatment for your child. Even if your child is on a clinical trial in the Children's Oncology Group or SIOPEN, your doctor will always prioritize your child and will always treat you the way that needs to be done. And so that may be veering off the clinical trial if need be. That's a long-winded question of saying, how have we gotten to where we are? I think we continue to need to learn from each other, to share results. Sue's spearheading with Andy Pearson, the INRG database is just one way of bringing us all together to really try to come to consensus when we can and to continue to push the field forward. I don't know, Sue or Maya, if you want to add anything more to that.
If I could, Maya, I just was gonna add, I agree with everything that Julie said. I'll give you a concrete example. So for the longest time, the Memorial Sloan Kettering folks had developed an antibody and had been testing it in an institutional study and providing antibody to the patients that were going there and showing in their institutional study that the antibody appeared to be effective in some of the patients. However, so that was a very important study and a very important observation. However, it was this children's oncology group who then said, well, let's test this to see not only is it feasible to give and safe to give, but does it lead to better outcome? Because that's really the question we want to ask and answer. And that's when we, and I took part of this way back when I was leading the disease committee, but we developed a randomized study where we had, as Rose showed you the results of, half the kids received immunotherapy and half did not after completing transplant. And we were able to show definitively that yes, adding antibody in this setting led to an improved outcome, and that ended up here. So I think with some of the institutional studies that are ongoing that are a single arm, you can comment about feasibility, you can comment about a little bit about response if there's disease to measure, and you can comment a little bit about how this very small group of patients did, but you cannot say, is it better than what is now considered standard of care? And so the only way to test that is a rigorous clinical prospective study. And in fact, in response to the paper that Ro had mentioned that was written by our patient advocate group, Andy Pearson and I actually wrote a response, and we said, we understand exactly what you're saying, and what has to be done is this has to be asked in a robust clinical setting to see if indeed we are able to utilize a different type of therapy that'll end up being as good or better than transplant and less toxic. But we can't just guess. We can't just wave our hands and say it's the way to do it. It needs to be studied prospectively, very carefully and rigorously. And then you get an answer, and then you can move on, and then you can have practice-changing decisions based on facts and data.
And I guess one more thing to add to that is that when we ran the trial looking at 2 transplants versus 1 transplant, we actually looked at, of the patients who got immunotherapy, did tandem transplant continue to improve the outcome? And the answer was yes, both for risk of recurrence and for overall survival. So it's really important that we study this, as Sue just said, in the right way to really know who can we, or can it be all children with neuroblastoma, remove transplant.
Yeah, thank you very much for this response. This covers another part of questions, and I think the fact that you have said the only confirmation to know whether something makes a difference, and Ro has very well underlined the necessity of taking into account what makes the change in the treatment strategy, has to be evaluated prospectively in a randomized way. And one of the questions was also how to prevent relapse. And what is the position of COG and SIAPEN towards the FMO? So this is another part that has been also discussed based on results that are single arm, not randomized trial, where we have to listen to parents and, of course, to respect results that are there, knowing that they do not come from a randomization. Can you complete the answer to this point very shortly?
Go ahead, Sue.
Do you want to go first, Julie, or you want to go first?
No, go ahead.
Okay. So yes, this was initially a single-arm study, and it was an incredibly difficult study to make any kind of conclusion with the initial data because these patients were in remission, and they were in remission after receiving standard upfront COG treatment, including immunotherapy. And as Julie showed, the outcome of those patients is quite good. So if you take a good group of patients who have already gone through all of their treatment and they're in remission, and then you give them another drug, there is nothing even to measure in terms of response because they're in remission. And so to say, when you added the drug or you didn't add the drug, it made a difference in terms of their outcome, it was impossible to interpret. And a number of us were really concerned about the way the trial was designed and how difficult it was, in my opinion, impossible to make any conclusions about DFMO. What ended up happening was eventually the company that ended up eventually licensing this drug ended up identifying what was called an external control group. And a very unique situation, all received antibody therapy on the ANBL-032 trial. So they had a control group of patients who were enrolled on this antibody study, who then went on to get DFMO. And they had another group of patients who were enrolled on this antibody study who did not get DFMO. And what they ended up doing was they tried very hard to compare the outcome of the kids who were enrolled on the study who did not get DFMO to those who did. And they did some statistical analyses trying to make sure that the patients matched in a variety of different ways. It was not the gold standard randomized control study. There are lots of folks that have debated about the robustness of the statistical analyses, but the way that it was done and the propensity scoring that was done, they presented all of this to the FDA, and the FDA decided that the data were sufficiently promising to recommend approval. And I believe the plan is for our upcoming trials, now that this is an FDA-approved drug, post-immunotherapy, this drug will be added to the clinical trials. But I just want everybody to recognize this is still debated among various statisticians in terms of how robust this type of analysis is, but it was sufficient for the FDA to approve the immunotherapy.
Thank you very much. Just to add, the same situation is in Europe. It will also be added and recorded in this way, but we are just a little bit behind with approval. But just to know that the same way of thinking is also within the CIPEN community in Europe. And maybe the very last question, this concerns maybe Julie. There is the question how to have the genomic analysis performed in your own child with neuroblastoma, which is the way to do it, how to get precision medicine, access to precision medicine, and especially whether what is the role of MET mutations, and are there studies ATRX? You see, quite a lot of questions, and there are many more, but we will not be able to maybe to discuss, but this is another topic.
Yes, yes. Well, in North America and in Europe now, a genomic analysis at the time of diagnosis is becoming much more common. In North America, through the Children's Oncology Group, there's actually a whole way of, through a clinical trial, for patients to have their tissue analyzed and their doctor to get that back, because it drives, if there is an ALK mutation, where how the patient will be assigned. The use of circulating tumor DNA is newer. There are ways to have that approved by insurance, but more often or not, it's part of a clinical trial. And we are working with a NANT, and I know my colleagues in Europe are also working to, again, study this, prospectively along the way, so we can really understand a lot of things about what's the cutoff, what really predicts that this is true tumor in your patient, in your child. These tests sometimes can have so much sensitivity, but they can sometimes be false. And so we have to be able to really follow this closely, track this with imaging to be able to understand what we're learning about these patients. And importantly, it also gives us an opportunity to learn how the cancer changes over time. And that will be really important as we think about what drugs we want to combine together. At this point in time, we've been very successful in targeting, or not, at least identifying a potential way to target ALK aberrations. We've known NMIC was a driver for years, and we've yet to find the perfect drug to target NMIC amplification. And similarly, there are other pathways like telomere and maintenance ATRX that we don't yet know exactly molecular targeted agents that can shut down those pathways. So I think more to come. We are trying to look at that, certainly in the NANT, certainly in other early phase clinical consortium like the ITCC in Europe and the PEP-CTN in North America. But right now, we have not been able to find the best agents for those. We did have one, we have an international collaborative meeting that happens every few years where we bring together investigators to talk about how we should prioritize drug development in neuroblastoma. And we recently had one in Boston a little over a year ago, and that should be coming out as a manuscript as well. And that helps us to work with drug companies to try to prioritize drugs that we want to be evaluated in neuroblastoma.
Thank you very much. And I think that we can end with that, to thank you again for being here, participation, presentation, and to say also to the parents that we are all like a big community, US, Europe, and other parts of the world, the importance of being part of trials, of protocols, because within these groups, access to new treatments are possible, access to new analysis. And this is why it's so important that we are really part of it, and that we invite also patients and families to participate in trials that will bring us the necessary knowledge and progress. Thank you very much. And I will hand over to Einat.
I won't take much of your time. I just want to thank you all for participating and for being with us. This is the first session out of 12, and stay tuned because we have a great program for the entire year. It will be every 5th of the month, unless it falls on a weekend, and then we need to amend the date. But thank you so much for your time, both. But whether you're a medical profession, thank you for your time. I know how precious it is, and also for parents and anyone else with the interest to support this vital cause. I appreciate everything, and we'll see you soon. We'll see you next month.
Thank you so much for having us.
Thank you.
Bye.
Bye.